So this short video is to introduce the disease of cancer to junior seminar because most of your papers are centering on nutrients in cancer. And so cancer is a very broad disease class. Not everybody's cancers are the same, even for the same type. And so what I want to do is introduce you to some of the molecular mechanisms that you might hear in the background of your papers. And know that this isn't a comprehensive background. We're just laying a foundation for you to be able to proceed. So, looking at some histological slides here, just from a visual perspective, cancer cells are different than normal cells. If we look at normal tissue, and let's look at this colon tissue example, the cells are very, very organized. There's a definite structure and shape to the cells, and the cells are each all very, very similar. If we come down to the colon cancer tissue, you can see that the cells are actually larger. More of the nucleus is taking up the cell, and we don't have the same type of organization. So cancer cells do not function the same way as normal cells do. And so as they're not functioning properly, they're not going to be able to help the integrity of the tissue to do its function for the organ and then the body. So cancer cells disrupt tissues. Here again we can see this is epithelium. If you look, here are the layers of the epithelium. There's this nice basement membrane that creates a border, and then below it we have connective tissue. Well, as we go through the stages of developing into a cancer, you can see the histology and the morphology of those cells change. Here it's breaking through that basement membrane. The cells are staining differently because their nuclei are much larger, taking up more of the cell, and there's more of the cells in this last example of the carcinoma. So what is cancer? Cancer is invasive and uncontrolled cell growth, and the cells that are growing are not normal. Each cancer is different. Cancers are caused by genetic mutations. Those can be familial or inherited mutations in the DNA, but most cancers are actually sporadic, where environmental, lifestyle issues, or chemical exposures mutate the DNA, and then those mutations are inherited. So even though you might have 2 patients with breast cancer, the actual underlying genetic mutations that have caused the cancer could be different in those 2 patients. So when we talk about cancer, we're dealing with a genetic disease, and by genetic we mean the problem is initiating with the DNA. And so there's 2 types of genes that we label or name because they help us to regulate cell division. And so the first are oncogenes. So oncogenes are genes that their products or their proteins lead to growth of cells, survival of cells, or they tell cells to proliferate. And so the common types of oncogenes or proteins that we have, these are growth factors. They could be receptors that bind to growth factors, proteins called mitogens that drive cell division forward. They could be transcription factors that once they are made, they are transcription factors that go and bind to genes that then are upregulated to help drive cell division forward. Tumor suppressor genes are really sort of the opposite. These are here to inhibit growth or division of cells, or even to promote apoptosis or cell death. Tumor suppressor genes are sometimes called the gatekeepers of the genome. Tumor suppressors are almost like little cellular machines that scan and look at the actual DNA bases themselves, and they make sure that adenine is across from thymine, that guanine is across from cytosine, and they can pick up on if there are mismatches or mutations in the DNA. if they find DNA damage or mutations, they aim to stop cell division and to try to correct the damage that's in the DNA. So when we think about what oncogenes and tumor suppressors do together, they're there to maintain the cells and to help them to divide and not pass on genetic mutations to their daughter cells. So we need both of them to keep tissues intact. However, when cancer occurs, we see mutations occurring in oncogenes and tumor suppressors. So in cancer cells, we see similar oncogenes get turned on. So they get upregulated or they're overexpressed in the cells. And if you look at the gas pedal and the brake pedal here, so the gas pedal is sort of like an oncogene. When you press the pedal to the floor, it drives that car forward. And in cancer, the oncogenes are in overdrive. They are pushing cell division forward. Tumor suppressors are an off switch. They tell the cell not to divide. In cancer, what we see are tumor suppressors. They actually lose their activity. So we see a loss of the tumor suppressors or a downregulation of the tumor suppressors, or the tumor suppressors get mutated in a way that they're no longer functional. And tumor suppressors are like the gas pedal. They tell the cell to stop dividing. So what we see in cancer then is this combination of oncogenes put the pedal to the metal and they tell the cells to divide, 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 divide. And the tumor suppressors that used to be there and say stop dividing, correct the mutations that we have, are no longer there. So now a cell can divide, and with it, it keeps dividing and replicating its DNA mutations to the daughter cells so that they have that same growth advantage or those same types of mutations that will then tell them to divide in an uncontrolled manner. So to look at this more specifically, here we have an example of how oncogenes work. So if we look over here, this is a normal cell. Normal cells to divide have to get input from surrounding cells, or they get input from cells at distant areas. So cell division starts with growth factors that bind to membrane receptors. They send a signal transduction message through the cell, and eventually that cell will turn on a series of proteins that are going to tell the cell to divide. And so we'll go through those steps of G1, S, G2, and then mitosis, and we'll have cell division. So the cell, or the proteins that are helping the cells to divide, those proteins, remember, that drive cell division are called oncogenes. So if an oncogene gets a mutation, so here's a proto-oncogene. Proto just means before. So here's an oncogene before. If it picks up a mutation that is going to make its product a mutated protein that might be always turned on, or it might make more of the oncogene so that there's more signals that say divide than normal. Now that oncogene is going to drive this cell division, and it will keep driving it. And as it drives it, each daughter cell is going to have that same genetic mutation that the original cell had. And these cells, because the oncogene is driving cell division, the cells no longer need input from a growth factor to divide. If you look at the left-hand side here, I listed some oncogenes for you. You can see, so these are their genetic names, the names of the genes, but you can see the types of oncogenes that you have. They promote cell growth. BCL2 blocks apoptosis. MYC is a transcription factor that's going to promote DNA synthesis. So if DNA is synthesized, that drives the cell then to divide. EGFR detects growth factors, so if we have more of those membrane receptors around, it's more likely that the cell is going to divide. We also have proteins that inhibit tumor suppressors. So MDM2 actually inhibits a tumor suppressor called P53. So if we have more MDM2 around, we inhibit tumor suppressors from doing their job. Here's an example of a signaling cascade, and it looks overwhelming at the beginning, but if we start to break it down and look at it, we can see different proteins. And in a cancer cell, many of these different types of proteins can start to have mutations in them. So if we start at the beginning, here's a mitogen. So it's a growth factor. Starting right there, if that mitogen is a protein, let's say that gene that makes this mitogen is mutated, or maybe we're making more of this mitogen than necessary. That can drive cell division. So the mitogen binds to its receptor, and that signal is internalized. The signals that drive cancer generally are what we call tyrosine kinases. That's a type of protein. But they bring that signal in, and at the end of the signal transduction pathway, normally there's some type of gene regulation or change in gene expression. And so that mitogen tells the cell to turn on genes that are going to start to drive the cell cycle, to push the cell to divide. And so here you can see one of those is a gene called MYC. So the MYC protein gets made, and MYC's job is to start to activate what we call cyclin CDK complexes. Those are necessary to begin to have the cell drive through and to start DNA synthesis, or S phase. So any of these genes in the pathway that help us to move towards DNA synthesis would be oncogenes. Tumor suppressors, remember, tell cells to stop dividing and to try to fix any mutations that there are. So if we look, here's a cell, remember, this was our defective oncogene. Now, if this oncogene is there because there is a point mutation, because there are mismatched base pairs, whatever that may be, our tumor suppressors are present in the nucleus, and they scan the DNA between, they scan it in G1 and G2, so before we go through mitosis. And if they pick up and if they see any damaged DNA, they literally tell the cell stop, right? They stop cell division in its tracks, and they assess the damage to the DNA, and then they start to make decisions. So the decisions that tumor suppressors can make is, first, they can try to fix the damaged DNA, and if they can fix it, then mitosis will resume and the cell will divide. They can say that the cell just should not finish its cell division, and they hold it so it gets put into like a permanent G0 phase, and that is called cell senescence. Or if the damage is so bad that the cell is afraid that mutation would cause cancer or cause a tissue disruption, then the cell can undergo apoptosis, or programmed cell death. So if we look at the types of tumor suppressors we have, over here, BCRA1 and BCRA2, those stand for breast cancer 1, breast cancer 2. Those are actually genes that encode for proteins that repair the DNA. Angelina Jolie made BRCA2 very, very popular this year because that was the gene that she was screened for, a mutation in it, and that's related to certain cancer risks. P53 is a transcription factor that when P53 knows that there's cell damage, it upregulates other genes that cause cell cycle arrest or apoptosis. And RB is an important protein that actually halts cell cycle in G1. If I go back to the previous picture here, you can see here is the RB protein. And so what RB does is, as the signal comes through for cells to start to replicate their DNA and start to divide, part of this cell signaling is actually to inactivate this RB protein. Once RB is inactivated, that frees this little protein that it has captured. And once that little protein is free, it upregulates genes that push the cell cycle through, and we have DNA synthesis. P53 has sort of a different job. So P53 will be activated when the cell senses either hypoproliferative signals, DNA damage, telomere shortening, which means the cells are dividing and at the end of their what we would call genetic life, or if there is hypoxia or some type of environmental issue that might be detrimental to the cell. So P53 becomes active, and once it's active, it upregulates genes that arrest the cell cycle, that cause senescence, which is a halted cell, or apoptosis. So here you can see a little bit of how P53 does this. So up here we have damage to the DNA caused by X-rays, it could be caused by UV light, it could be caused by certain drugs or environmental exposures. And so here again, we start to upregulate a series of proteins if you look in this picture. And each of these series of proteins, these are potential tumor suppressors because they are working to stop the cell from dividing. So as the signaling cascade comes through, eventually it activates P53. P53 is going to turn on a gene called P21. So P21 is another tumor suppressor gene. And P21 sequesters and stops the activity of cyclin CDKs so that they cannot tell the cell to synthesize its DNA. And if a cell is not synthesizing its DNA, it's not going to divide. So that is just a brief intro to cancer. You will see many of these tumor suppressors and oncogenes in your papers as we review how nutrition affects cancer. If you have any questions or any questions as we go along in the class, please let me know.